All right, so on to chapter 2. Chapter 2 is about the chemistry of life, and although we're not going to spend a lot of time talking about chemistry in this class, we can save that for your chemistry class if you need to take it. But it is still important to understand some basic concepts of chemistry because of how integral it is to understanding biology. Before we get into that, a couple of quick review questions, some types of information you should have already gotten from chapter 1. So, and you can always pause this if you want to think a little longer. But question 1, knowledge that covers general truths or the operation of general laws, especially when acquired and tested by the scientific method, is referring to what? Science. Biology is science, basically. You have to follow certain principles when you're studying things, and science is the knowledge that covers that. A suggested explanation for an event that can be tested. And again, it's important from a scientific standpoint that the suggestions that you come up with have to be able to be tested. So this would be a hypothesis. Also from chapter 1, it's a type of diagram that shows the evolutionary relationships among biological species based on their similarities and differences in both genetic and physical traits. And this refers to a phylogenetic tree. And again, if you want to take a little longer to think about the question, you can always hit the pause when the question comes up. So the part of an experiment that doesn't change during the experiment. And again, this is another integral, important part of science, is that you always have a control. If you were a drug company and you were testing the efficacy of a drug, you would give the actual drug to a group of people in your experiment, but you would also give a placebo, something that should not help them, to another group of people. And those would be the control people or the control group, the part that doesn't change, that isn't affected by whatever the experiment is. Also from chapter 1, the smallest fundamental unit of structure and function in living things. Smallest fundamental unit. Now again, this isn't referring to the very smallest particle, but the smallest fundamental unit of life is the cell. Again, yes, there are smaller things, and we're going to talk about those today, but they're not considered functional units of life. An atom of carbon isn't life, isn't a living thing. And finally, all individuals within a species living within a specific area, like this group of penguins in the image. Those individuals that are in the same area at the same time are called a population. All right. So, and again, I'll maybe be referring down to my book on occasion to make sure I keep track of where I am. This class basically is going to start with the small stuff, atoms and molecules, and then we're going to put molecules together to build a macromolecule, such as what we're talking about on this slide. The food on that cutting board are rich sources of the macromolecules that our bodies need to be able to process energy and then provide everything that our bodies need to stay alive. And so then we're going to build from macromolecules to the bigger and bigger things until the very end of the semester, when we're going to be looking at larger aspects of the earth, populations of living things, communities, ecosystems, the earth as a whole. And so again, keep in mind, we're just kind of building from microscopic stuff through the macroscopic stuff, the stuff that you can actually see. So there's a link here to a video about sourdough, how sourdough bread is made. So I'll give it a second here to load. There are links on the videos, links to videos, I should say. And in some cases, I may just direct you to them, and in others, I may actually play it like I am here. back to the slides here. I've seen that video a whole bunch of times, so I bowed out there for a few minutes. But basically, I just show that as an example of how much we do depend on science for everything, including our food, making things like bread, depending on single-celled organisms, yeast, and understanding how all of that's necessary to make something like a sourdough bread. And again, we'll build on this, and we'll start looking at the actual macromolecules, things like carbohydrates and proteins, in the next chapter. But first, again, a little bit of chemistry. So we mentioned in the previous slides, there was a question about the smallest fundamental unit of life being a cell. Of course, you probably know there are things that are smaller than cells, atoms, for example, which is what we're going to start with here. And atoms are actually made up of smaller particles themselves. So we have protons and neutrons that are smaller particles of an atom that are located within inside of the nucleus of the atom. And the neutrons are actually a neutral charge. They're not positive or negative. Protons, pro-positive, have positive charges. And then we have electrons that are orbiting around the nucleus. And these electrons have negative charges to them. And we'll get into a little bit more about the relevance of positive and negative charges. I'm not going to go to this link, but again, while you're watching this, you can always take a little break from me and click on the different things that are listed here and go off and watch a video that's off of YouTube. Now again, this isn't a chemistry class, so I'm not going to make you memorize a bunch of details about the atomic chart or you know, the different elements and things, but still there are a few things that I think are important enough that we spend a few minutes on them. So characteristics of atoms, for example, every atom has an atomic number, and we'll look at the periodic table in just a moment. And on that periodic table are going to be things like the atomic number, which is the number of protons that are found within a given atom. And then we also have what's called the mass number, which is the protons plus the number of neutrons. And then we can also determine the number of neutrons if we've got the mass number and the atomic number. Because if we subtract the mass number from the atomic number, then obviously what we're left with are the number of neutrons. And basically the reason that we even care about number of protons, neutrons, electrons is that it provides us with a little more information about how elements can react with each other when they're combined with each other. And so we'll look at a couple examples of that coming up here. Another term to be familiar with is the word isotope. And so isotopes are other atoms that have the same number of protons but a different number of neutrons. For example, your book talks about carbon-14 and carbon dating on page 35. And so normal carbon has 6 protons and 6 neutrons for a total of 12. But then carbon-12 can also have an isotope, carbon-14, which has 6 protons and 8 neutrons. And the evolution in action inset in your textbook, again on page 35, talks about carbon dating, using the properties of elements like carbon to be able to date objects, fossils, for example, things that are maybe tens or hundreds of thousands of years old. There is limits, though, to what carbon-14 dating can be used on. If things are less than, say, 50,000 years old, then we can use carbon dating. Things that that are older or even much younger, we might have to use a slightly different process for. But basically, carbon-14 dating is a way that we can date things that were once alive. So you can't date a rock, for example, or some inorganic object. It has to be something that was alive. And the reason we're able to do this is because these isotopes decay at a measurable rate. And so we can measure that rate of decay, basically, and then that allows us to determine the age of something. So here's that dreaded periodic table of elements on page 34 of my book, and I realize there are occasions when there are maybe like 2 different versions of this textbook out there, and the page numbers may be slightly different from each other, but the content's the same, the figures are the same, they just may be a few pages off. But again, on my book, page 34 is where you can find the periodic table of elements. And again, fortunately, this isn't chemistry, so you're not going to have to memorize this table. But I do want to highlight here that if you look at any one of these individual elements, like hydrogen, which is atomic number 1, that you'll see the symbol, the standardized symbol for this element, the relative atomic mass, and then the full name of the element. The periodic table is also color-coded, and the color codes are explained here. Again, I'm not really going to worry about getting into you know, what an alkali metal is versus a transitional metal. We'll save that again for your chemistry class if you end up taking it. But so here is a sorry. We see hydrogen here depicted with the protons and the neutrons and the electrons. So hydrogen has 1 of each. And then as you start moving up the table, you get to things like helium, which has 2 protons, 2 neutrons, and then 2 electrons orbiting outside of the nucleus. And then again, you can use the information in each of these squares to again, look at the atomic number, and that can again tell you about the overall kind of structure of that element. So one of the things that's relevant from a chemistry standpoint to our study of biology is the way that these different elements form bonds with each other to form larger molecules, or what will eventually build on molecules and then macromolecules. So on my page 36, we get into chemical bonds. So what we have here are, keep losing my mouse. I don't know why I can't get my mouse back. We have here in the purple color, the Na is a sodium atom, and the sodium atom has 2 electrons in the orbit immediately around the nucleus. And then if you count them, really wish I could get my mouse back. See there. Okay, and my mouse is gone again. Anyway, if you count the number of electrons in the first orbit, the circle closest to the sodium atom's nucleus, you'll see there are 2. And then if you go in the next outermost orbit around the sodium atom's nucleus, you'll count that there are 8 excuse me, electrons. And then with sodium, there's one more electron in the third most outer layer. Elements like to be stable, and so they like to fill their outermost shell and have it full whenever possible. Now, full means 8 electrons. So the innermost shell can only ever hold 2, but then any other shell beyond the innermost one is full, or it can accept up to 8 electrons. So again, if you look at the sodium element there, there's 1 extra electron in the outer shell. When sodium comes in proximity to chlorine, which is indicated by the Cl element to the right, notice if you count the number of electrons in its outermost shell, there are 7. So when sodium and chlorine come in close proximity, the outermost electron tends to want to go over to the chlorine. That way, both the sodium ion has 8 electrons in its shell, and then now the chlorine ion has 8 electrons in its outermost shell. What happens then is when sodium gives up an electron, we refer to it now as a cation. Because if it's losing 1 negative charge, basically, it's giving up a negative charge, that means it's going to have a net positive charge. So the sodium, notice down below, there's a little plus sign there that indicates that sodium cation now has a slightly positive charge because it gave up its outermost electron. The chlorine then, because it accepted that outermost electron from the sodium, it's now happy because it also is stable, but it picked up another negative charge, so it overall now has a negative charge, the element as a whole. And so we refer to those as anions. So cations have slightly positive charges because they give up electrons. Anions, like chlorine, have a slightly negative charge because they accept electrons. And so whether you donate or accept electrons, basically this process is called electron transfer. And what happens then is these 2 molecules now are bonded together by what are called ionic bonds. So these are types of ions, a cation and an anion. Sodium chloride is salt, if you weren't aware of that. And so then this sodium and chlorine ion are held together in what's called an ionic bond. So understanding chemical bonds is important to biology. Another very important type of bond is the covalent bond, and it's the most common type of bond that we see in molecules, and it's also the strongest bond. Ionic bonds are relatively strong, but they can be broken without too much energy. Covalent bonds are very strong, and they don't just break apart in water like ionic bonds could. So not only do we have covalent bonds, but they can either be polar or non-polar. A polar covalent bond, such as we would find in the case of a water molecule in the upper left of my screen there. Trying to figure out here why I cannot get my pointer. So the polar covalent bond in the upper left of the screen is a water molecule, 2 hydrogen and 1 oxygen. And so that would be a polar covalent bond. And it's polar because the electrons are shared by the atoms, but they spend more of their time closer to one of the nuclei. And so water ends up with a oxygen that has a slightly negative charge and the hydrogens each that have a slightly positive charge. But the electrons, even though they're shared by the oxygen and the hydrogen, they're spending time closer to one of the nuclei than the other element. Sorry. And then a non-polar covalent bond basically shares the electrons equally. The electrons aren't favoring one molecule over the other. So the molecule in the middle is methane, carbon with 4 hydrogens. And then the other example there is oxygen, 2 oxygen molecules, where again the electrons from those oxygen molecules are being shared equally. They're not favoring one molecule or the other. Third type of bond that we mention is the hydrogen bond, which is a relatively weak interaction between, in this case, we have water molecules. Again, you can see 3 water molecules there. And the hydrogens are being held to the oxygen by covalent bonds, like we saw in the previous slide. But then the water molecules themselves are being held together by these hydrogen bonds. So that's, again, an interaction between the positive charge of the hydrogen ion of one of the water molecules and the negative charge of the oxygen atom of another molecule. So you can see the arrow there, or the line showing you where the hydrogen bonds are versus the covalent bonds. Hydrogen bonds also are important because they hold our strands of DNA together. So again, basically hydrogen bonds are what allow there to be bodies of water. Why you can put water in a glass, or why water stays together in a lake. But it's also why water can evaporate, those bonds break, individual water molecules get separated, and because they're so small, they can float back up to the atmosphere until they start bonding together again, and you get enough water molecules all stuck together with hydrogen bonds so that it's so heavy that it can't stay up in the atmosphere anymore, so that causes it to fall to the earth. So aside from the bonding that we're talking about here, water itself is kind of a really interesting molecule. It's basically what allows there to be life on Earth. And so, again, it makes it kind of relevant why we should understand how water is made, covalent bonds between hydrogen and oxygen, and then those other bonds that hold all the water molecules together. We also have substances in the environment that are non-polar, such as oil. Again, maybe going back to the cooking idea, you've all heard the idea of something about oil and water don't mix, kind of referring to like 2 things that are very different just shouldn't be put together, or they can't be put together. And that's why water won't dissolve something like oil, because the hydrogen bonds don't readily form in a non-polar compound, and we refer to that as being hydrophobic. Hydro, water, phobic, as in not liking or not pairing with it. So you make some sort of vinaigrette, you mix oil with other things, and you stir it up, but you can see the little globules of fat kind of staying suspended like this picture. You can mix them up and use it, and it's there, but the oil never is mixed or dissolved into vinegar, for example. And this also comes into play with things like pesticides. There are a lot of pesticides that are non-polar compounds that don't readily dissolve in water when they're sprayed on the environment. Much later in the semester, we'll talk about things like biomagnification, the process where some of these chemicals can get in the bodies of living things. And because they aren't water soluble, they accumulate in the fatty tissue inside of things like us or salmon, and they don't break down readily because, again, of the chemical properties that we're talking about here. I mentioned a moment ago that basically water is one of the most important things on this planet. All living things are composed mostly of water. But beyond that, the Earth is the right temperature. If it were much colder, even if we had water, it would be ice all the time, right? And we know we have places on the planet where there's lots of ice or where the precipitation falls as snow rather than rain. The lattice structure of ice basically is what makes it less dense. So when you have icebergs, they float, they don't sink. And that's because when water gets chilled below 32 degrees, when it freezes, it forms this sort of structure that you see up there. And basically the molecules are spread out and there are spaces in them, so they aren't as dense as when they're liquid water. And so again, that's what allows ice to float, less dense than water. We also have temperatures on the planet where it's warmer. And so liquid water can actually evaporate and become a gas. And then that gas creates things like humidity, wetter feeling, sticky. But then that gas also goes up into the atmosphere, hangs out in the atmosphere, and then eventually if it condenses like we were talking about, the individual water molecules bond with others, they get bigger, heavier, and then they can fall back to the Earth. The other thing before I go on is big bodies of water have a lot of water in general has a lot of other properties that are relevant and important to life. You probably maybe have all thought about if you go over to the coast, it's always cooler there. It's never as hot or never as cold as it could be away from the ocean. That big body of water that the ocean is moderates the climate. Not only does it moderate just the temperature of a specific town, but because of how big the oceans are, they moderate the climate. They don't let an area get really hot or really cold because water has the ability to absorb a lot of heat, a lot of energy. And then that, again, keeps the temperature from getting really hot or really cold. So basically, it's able to stabilize temperatures and create lots of different climates that we have around the planet. Water is also a really, really good solvent. In other words, it's able to dissolve other substances. That's what a solvent is, sorry. And we were talking about sodium chloride a moment ago. When you have chlorine and sodium come together, they exchange electrons to become stable. Again, chlorine ends up with a slightly negative charge because of that. Sodium ends up with a slightly positive charge because of that. But the other thing that happens is, again, these are being put in water, right? So not only do you have the chlorine and the sodium ions, the anion and the cation, but when they're actually put in water, so you put some salt in the water you're boiling for pasta, that salt basically dissolves because the water is a good solvent. The reason it's able to dissolve, or what dissolve means, is that you get these spheres of hydration that form around the sodium and the chlorine that's in that table salt. So you see all the little molecules of water there. From the top down, you put table salt in water, and then what happens is the water molecules start attaching, and we'll look at a little more of that. But remember, the chlorine ion has an empty shell, right? Once it's given up that electron to sodium, it's got an empty outer shell, right? It's got 8 electrons in one shell, and then the next shell is completely empty. So what it's able to do, because it's completely empty, is attract positive charges. So you see the bottom row there, the chlorine ion now has 4 water molecules attached to it. If you count, there are 2, 4, 6, 8 hydrogen attached to the chlorine. Remember, again, you can have 8 electrons in the outer shell. So basically, the 8 electrons from those hydrogens are able to bond with the chlorine. And now, because that chlorine is surrounded by water, it is dissolved. And the same thing with the sodium. The only difference with the sodium is, notice that it's the oxygen that have attached to the sodium rather than the hydrogen. Again, that's because, remember, the oxygen has the negative charge in a water molecule, and it shows you that up in the upper left there, or the upper left center. And if you were to look at a sodium, it's got empty spaces as well out there in the outer shell. And so now it's attracted, the negative charge of the oxygen is attracted to that slightly positively charged sodium. And so it also gets surrounded by 4 molecules of water, which then allow the sodium to dissolve. So dissolving really means these spheres of hydration form around the element so that it is able to be dissolved into water. Water also has a couple other properties, cohesion and adhesion, that create some interesting things. So first, just think of cohesion as meaning the same, that the same things attract each other. Adhesion meaning different, and we'll look at what that means. So cohesion is basically what would allow water, little droplets of water to form. One of these droplets of water there on that leaf of a plant, that droplet, any one of those droplets, and notice there are different sized droplets, are not all the same size, so that's not just one molecule of water. That's a bunch of molecules of water that are being held together by cohesion. So being able to form a droplet of water, or any time you have water that kind of stays held together, would be through cohesion. The other thing that I know we've all observed at some point, you've got a straw in a drink, like these glass pipettes in this liquid, and you can look and see that the water level, or soda, whatever you've got, is actually going up the straw a little bit. And the smaller the size straw, the farther it goes up. Now notice on these pipettes, and again, I do not know why I lost my mouse pointer here, but notice the pipette has a smaller hole inside of it. The tube on the far right has the smallest diameter hole, and if you follow the liquid, it goes way up that pipette. So we're not looking at the liquid right down at the water level, we're looking at it up way inside of the pipette. The middle pipette has the largest diameter hole, and the water goes the least farthest up inside the tube because of the larger size hole. But the fact that those water molecules are being held together by cohesive forces, and that they're being forced into a small space, and that the water molecules are kind of grabbing on that opening, would be the process of adhesion. The water molecules grabbing onto the side of the glass pipette. Whereas again, water molecules grabbing onto other water molecules would be cohesion. Water also tends to have a certain amount of surface tension. It's the reason why if you dive high enough off of a platform, if you belly flop, it's going to hurt a lot more. It's going to make a lot more impact than if you dive and go in like a diver does with a narrow point, right? Your hands going in or your feet going in, not belly flopping. Sometimes they talk about like when you're water skiing, if you're going really fast and you hit the water, it's kind of like hitting concrete. It's because water has that surface tension and it has resistance and a lot of other properties, but part of it is the surface tension. And it's basically that the water's pulling down at the same time that the surface tension is pushing back up. So you can carefully set something like a needle, which is metal, right? Kind of heavy for its size, but you can carefully set it on the surface of the water, as long as it's sideways, and you can get it to float. Of course, if you take that same needle and you try to drop it in the water this way, it'll sink to the bottom because there isn't much surface area compared to when you have it oriented in this direction. So this allows, for example, you've seen maybe water striders, different kinds of insects that can scoot along the surface of a pond, and even ducks, when they take off, they can kind of run along the surface of the water while they're flapping their wings to get up enough momentum to actually get airborne. So general properties of water, some real basic information about the atomic numbers of elements and the periodic table. Again, you're not going to have to memorize the periodic table or any specific information about elements. There could be a question or two on a quiz about elements and how they bond together. And those are always questions that you'll have some time. You could, look those answers up in the book if need be. All right, so this would be a good spot to maybe take a pause before we move on to the next topic. So I'm going to go ahead and pause for a moment. All right, we'll get back with it. So the next topic, chemistry-related topic in the book, deals with buffers, pH, difference between acids and bases. So I'm sure you've all heard of pH, may or may not kind of understand what it actually is. We measure pH on a scale of 0 to 14, where 7 is actually neutral. If the number, the pH is smaller than 7, we consider that something that's an acid. If the pH is higher than 7, we consider that a base. What pH actually is, though, from a biological or chemical standpoint, is that it's a scale that actually measures the amount of hydrogen ions that are in a substance. Hydrogen ions being designated by an H+. Plus meaning, again, that the hydrogen has a slightly positive charge. So, again, we may think of it just as being an acid or a base, but chemically it's the measure of hydrogen ions that are in a substance. And so then typically we measure pH by looking at how acidic or how alkaline something is. Again, acidic would be numbers pHs smaller than 7. Alkalines would be those that have a pH greater than 7. And so there's a figure 2.12 in your book, my page 43, that shows you kind of the range. Distilled water being 7, which is neutral, and then urine, slightly acidic, black coffee, and then we start getting into tomato, orange, lemon juices that are more and more acidic. And then finally gastric acid, which is the acid in our stomachs that helps break down stuff that we eat, being among the most acidic. Seawater is slightly alkaline, followed by baking soda, milk of magnesia, on up to bleach. Even soapy water is really quite alkaline. And bleach, probably obviously, more so you thought of that as being something that would be high on the alkalinity scale. And so again, we can use this information to understand how things are going to react and other chemical properties. But even more importantly, perhaps, is understanding pH is important because our bodies, living things in general, have a balance in our cells. Every one of our little microscopic cells, the billions or trillions of cells in our body, all have to maintain a certain pH level. If we drank a lot of lemon juice, although I don't know why you drink lemon juice, orange juice or lemonade maybe, but if you drank a lot of really acidic liquids, if our body didn't have a way of dealing with that, we would make our cells really acidic, and that would kill them. So our bodies have this built-in mechanism to help balance out. Whether we take things in that are acidic or we take things in that are alkaline, our bodies have a way of monitoring that, and that's through basically the use of buffers. And we'll look at what buffers are in just a moment. So again, lots of definitions. Science is, like I've mentioned, a foreign language basically. So important to understand the actual scientific or chemical definition. So acids basically are substances that provide hydrogen ions. So when you add an acid to a solution or to a cell in our body, that's giving hydrogen ions. And so what that does then is it tends to lower the pH of the solution that it's in. And remember, if the pH is less than 7, those are considered to be acids. So chemically, that's what an acid does. And then the stronger the acid, of course, the more readily it gives those hydrogen ions. A really strong acid is giving lots of hydrogen ions. So it makes the solution really acidic, and so forth. Bases then provide hydroxide ions, which are designated OH, and hydroxide ions have a negative charge. So notice the hydrogen ions have a slightly positive charge. Hydroxide ions have a slightly negative charge. And so they work the same way that acids do. They contribute, in this case hydroxide ions though, which causes the pH to raise. The stronger the base, the more it gives those hydroxide ions to the solution that it's in. Again, as I was saying, if our cells didn't have a mechanism by which they could take those hydrogen ions or those hydroxide ions and moderate them, it would be really easy to overload our body. Too much acid, we can make our bodies really acidic. That would kill them, our cells, it would kill us. Too much alkaline substances could create the same sort of problem, basically just throwing us out of balance. So we have buffers, which basically are there to be able to absorb those excess hydroxide or hydrogen ions. And then that keeps the pH maintained at the level that it needs to be at to be healthy and functional. These buffers are actually weak acids or bases. A weak base can kind of counteract some acids. Weak acids can counteract bases. If your stomach is unhappy, you ate something that didn't agree with you, or you drank too much coffee. And coffee is acidic. And if your body, your stomach gets unhappy because of all that acid, you can take something like Pepto-Bismol or milk of magnesia. And it's those chemicals in those nasty things that you drink that are basically bases that counteract the acid that you're putting into your body. So these buffers are weak acids and bases, and it's actually the cytoplasm that's the liquid inside of the cell. See all those little things inside that cell? We'll talk more about those in a few chapters. But the liquid that's in between all those little organelles is cytoplasm, and that's basically where the buffering takes place. In our body, carbon dioxide is actually a buffer. Now we know we can die from carbon dioxide, too much of it being inhaled into our body. But our body does need CO2 as well to serve as a buffer to help counteract the acids and bases that get into our body. If you're in the lab section of this class, each week we'll be doing one or more lab activities that allow you to kind of do some hands-on stuff, looking at, for example, week 2's lab, we look at buffers, and we can actually do some experiments to see how buffers work. So obviously not every single topic that we talk about in the book are we going to be able to do a lab activity on, but the labs, if you're in the labs do follow what we're doing topic-wise in the book, and so you'll be able to get some hands-on science with respect to the stuff that we're just kind of talking about in the book. Keep my throat lubricated as best I can. All right, the next section in the book, 2.3, deals with biological molecules. So we look at those elements like carbon and hydrogen and oxygen. When they start bonding together with covalent bonds or ionic bonds, they form molecules. So when you have one or two or more different elements that bond together, you're looking at making a molecule. On this planet anyway, carbon is the basic foundation or building block element for molecules in living things. Hang on a second, gotta pause. All right, sorry about that. Had to run and get the phone. I'm the only person home at the moment. So again, on this planet anyway, carbon is the basic building block of life. Carbon has characteristics about it that not all elements have. For example, it has 4 empty spaces in the outermost shell. So it can form 4 covalent bonds, which allows it to create a whole bunch of other molecules. Simplest is methane, C4, which is pictured there on the screen. But as we're going to start seeing here in the rest of this section, just basic elements, carbon, hydrogen, oxygen, nitrogen, those 4 elements, primarily carbon, hydrogen, and oxygen, form so many different things just based on the different ways they arrange together. And again, I'm not gonna make you memorize these chemical formulas or be able to identify molecules. Like when you look in figure 2.14 in your book, you're not gonna have to do much identifying those molecules, but it's still important to recognize just those 3 or 4 elements, and then all the different ways they arrange themselves make the huge amount of diversity of molecules on our planet. But at the root or at the core of it tends to be carbon, at least on this planet. So again, here are molecules, stearic acid, which, and all I'm going for here is not to memorize what stearic acid is, but see that one way that a molecule can form is with a long chain. So you have all these carbons bonded together and then other elements coming off of them. So they can form chains like stearic acid. They can form, kind of an element that has a whole bunch of other little bonds off of it. In this case, glycine, which is a component of proteins that contains carbon, hydrogen, oxygen, and nitrogen, those 4 basic ones I was pointing to. It can also form in rings, like many of our sugars. Glucose, the basic building block of life, the basic sugar, the basic food. Plants make it, animals eat it and break it down. You can see in that case, it forms a ring, and then it has the other elements coming off of that. So basically just showing you the different ways that carbon atoms can be bonded together to form molecules. So from the standpoint of nutrition, you typically look at things like carbohydrates, fats or lipids, proteins, and those, some of those basic molecules are the kinds of things, sugars, which are types of carbohydrates. You look for those things on like a label on a food item. And so because those are kind of the basic food items at a molecular level, we're going to spend a few minutes talking about those. So again, note there's a link here that you can click on and watch a short clip about biological molecules. If you want to do that, I'm not going to do it here, but just a reminder that it is there in the PowerPoint version of this presentation if you want to go and check that out. So again, the basic building block carbohydrate is glucose, and its chemical formula is C6H12O6. So there are 6 carbons, 6 oxygens, and 12 hydrogens. I've got my mouse now and I don't know what I did different. Notice that these are in a ratio of 1 to 2 to 1. 1 carbon for every 2 hydrogens for every 1 oxygen. And if you look at the actual visual arrangement, you can count these. The carbons are there, even though there isn't a C there. It's implied that there are carbons there. So 1, 2, 3, 4, 5, and then the 6th carbon is off there. You can count the 6 oxygens and the 12 hydrogens if you need to prove that that is indeed the correct formula. So glucose, again, is the basic sugar that plants make. They take the sun's energy, do the process of photosynthesis, which we'll talk about in a later chapter, and then they build these glucose molecules, which in turn are eaten by animals, which then animals like us use that basic carbon, hydrogen, and oxygen to build all of the other molecules that our bodies need for survival. Got a mention here of low carb diets so first of all, low carb diets where carbs are viewed as being evil, and it boils down to one simple thing, and you've probably heard this too, right? Everything in moderation. Our bodies need carbohydrates, so you do have to have a certain amount of carbohydrates in your diet, whether they're simple or complex. You do have to have a certain amount of protein. you do need to have a certain amount of fat. So all of those things are necessary parts of our diets. It's just a matter of if you ingest too much of the carbohydrates in sugars, like within soda, for example, or you ingest too much fat through eating too much red meat or too much butter, that kind of stuff. Our bodies need all of these things, and as long as you are doing it in moderation, then going kind of a low carb diet isn't necessarily a great way to maintain yourself over the long run. People tend to lose weight on low carb diets, but it tends to be a lot of water weight. In other words, it isn't really fat or anything you're losing if that's what you're trying to lose. And so then, as soon as you give up on the low carb, then that weight tends to come right back. Athletes use knowledge like this to their advantage. You might hear of like marathon runners that load up on carbs. They'll eat pasta, for example, the night before they run a marathon, or the morning of running a marathon. Carbs give us quick energy. They're not the sort of the slow burn, long-term energy. So our bodies, again, need energy right now that we can burn and utilize to do work, to move, to run, to think, to study, whatever you're doing. And again, a lot of that comes through our bodies breaking down carbohydrates. Not all carbohydrates are created equal. Again, here's glucose on the left side shown in a slightly different fashion. This isn't, again, exactly how it looks. This is just another diagram to show the orientation. Remember, it's actually a ring, not just a straight line. But sometimes we show the molecules this way just to be able to look at differences. For example, glucose, galactose, and fructose are all chemically the same. In other words, they have the same chemical formula. They're all C6H12O6. And again, you can count the number of carbons, hydrogens, and oxygens if you don't believe me. But what's different about these are the arrangement. So these are called isomers, or more specifically, isomeric, iso meaning the same, monosaccharides, meaning one saccharide, one glucose molecule. So isomeric monosaccharides. So glucose, galactose, and fructose are all isomers, and they're all monosaccharides. They have one basic they're made out of one basic sugar molecule, one basic carbohydrate, C6H12O6. But glucose, galactose, and fructose all have slightly different chemical properties. Fructose is sweeter to the taste than glucose is, for example. You've probably heard of the term of a high fructose corn syrup. Corn syrup. The kind of sweetener that we used to find in all types of sugary sodas. Again, too much of that is not good for you, but that's not to say that fructose is bad for you inherently. It's just a matter of how much you're ingesting. So again, more terminology: isomeric, monosaccharides. They all have the same formula, but they have different properties. We used high fructose corn syrup as the primary sweetener in the United States for a long time. And it was largely because we get it from corn, the name corn syrup, right? And basically corn was cheap. That's the bottom line. Talk about foreign sugar tariffs that made our sugar, our corn growing in this country subsidized, so it made it cheaper. And so it was the easiest way to make sweetener. And so we used it, and it's still used in soda and other food products. Again, it's not that it's inherently bad for you. The Food and Drug Administration says that high fructose corn syrup is not bad. It's okay. But, you know, if you drink 10 sodas a day, that's not good. It's the volume of it. Normal corn syrup, the kind of corn syrup you might use in baking, buy those bottles of corn syrup. We used to put red dye in there and make fake blood out of it. That's normally it's glucose. So it's not the fructose, it's glucose, still C6H12O6, but the fructose tends to still be much sweeter than glucose. We can actually, in your bodies, convert glucose into fructose because of enzymes that are used to convert the glucose to fructose. And we'll look at enzymes in another section. But it's basically that process that our bodies do. And fructose, again, is sweeter than glucose, even though it's the same chemical formula. There are other molecules, though, that are still carbohydrates. And we're still talking about carbohydrates here. Starch. Notice that starch is a chain of these molecules. And again, these are still basically glucose molecules. Each one of those circles is a glucose molecule. And starch, which is a carbohydrate, it's not sweet though, right? It's a different kind of starch, is formed by a chain of these glucose molecules. Glycogen has a little side chain shooting off of it, but notice again the core is still that glucose molecule. Cellulose. Anybody know what cellulose is? It's basically it's what makes up plant cells. Cellulose is what makes plants hard to digest, but that's still a type of carbohydrate. Chitin. So cellulose is what kind of makes plant cells hard, right? You can have a tree that grows up and it doesn't flop over because it has cellulose in every one of the little cells. In animals like crabs, shellfish, they have chitin. So instead of having a skeleton inside their bodies like we do, they have an exoskeleton. And that skeleton isn't made of the same stuff that our bones are made out of. Their skeleton's actually made out of chitin. And so you can see a little more complex formula there. But these are actually now called polysaccharides. A monosaccharide is one sugar, right? Just one sugar molecule. So you can see like starch, it's made up of 4 of these sugar molecules. But now because it's made up of multiple sugar molecules, it's a polysaccharide, mini saccharide molecules. Mono, one, poly, mini. And so again, just the arrangement of these sugar molecules, how they're bonded together, the order they're in, makes up everything from, you know, sweetener to the shell of a lobster. But those are all still carbohydrates. To put it in perspective, because a lot of times in science, when we're talking about things we can't see, it's hard to visualize how things are organized. So here we have at the bottom, and again, I lost my mouse again. I don't know why it comes and goes, but it does. We see strands of those glucose. So the little box there highlights one glucose molecule, a monomer, one. Those bond together to form cellulose. Many strands of cellulose molecules all twine together to make these microfibrils that you can see on the figure there. I don't think this specific figure is in your book, but it's obviously in the slide here for you to look at, or you can find these kinds of things online. But so all these individual glucose molecules bond together to form cellulose molecules. Those cellulose molecules all wind together to form what's called a microfibril. These microfibrils then are what make up cellulose, which are in the wall of a cell. You see in that image there, that little square on the top of the screen, it shows the cell wall around that green cell. And then all those cells form together to make something like a leaf or any other part of any other plant. So sometimes it's important to be able to piece together how all this stuff fits. So this is a good image to kind of show you from the individual molecule to the entire thing, like a leaf. And again, you see the chitin there, which also is the outer shell of insects, for example, arthropods in general. Again, we learn from the natural world around us and even find ways oftentimes to apply it to help us. Chitin is actually used now in surgery. So we artificially synthesize chitin. It's the same structure, the same formula that you see there. But basically it forms a really strong but flexible thread that can be used. And so if they're sewing up something inside of you, like they have to cut into your intestine to maybe get out an obstruction or something, they can sew it up with that type of chitin, and then it basically stays intact for some amount of time. And then as your body is healing, that thread starts to break down. And there's no need to basically go back and have to remove sutures. I know they use it in veterinary medicine too. So using molecules out there in the natural world and applying them in a way that helps us as humans. So that's carbohydrates. And again, a real quick overview of carbs. The next macromolecule are lipids or fats. Remember, we were talking about oil and water not mixing because oil or lipids are non-polar, which makes them hydrophobic. Water hating, hydrophobic. Hydrophilic means water loving. Hydrophilic molecules are attracted to each other. Hydrophobic molecules tend to repel each other. Whereas carbohydrates are short-term energy. Basically, you eat carbs and your body breaks them down and uses the energy quickly. Lipids are meant to be long-term storage, energy that's put into storage for later use. Things like polar bears that live in very cold climates depend very much on being able to eat a lot of really fatty foods, salmon, for example, in the fall. And they're just packing on lipids, fat. They're putting fat into long-term storage so that when they hibernate and they're not eating all winter long, those lipids now are slowly utilized by the body to keep it alive over the course of maybe weeks, if not actually months. And again, our bodies need lipids just like we need carbs and proteins and other molecules. It's just about moderation, everything in moderation. So beyond molecules being hydrophobic, the lipids that are actually in the fur of animals like seals or otters or polar bears, if they swam in the water and the fur absorbed the water molecules and the water got right up against the skin, that would cause the animal to get cold. I mean, we don't have much hair on our bodies compared to most other mammals. If we get in really cold water, the water's touching our skin instantly, and our body starts losing heat because of that. animals that live in the water, especially cold water, and even ducks have hydrophobic feathers. They live in the water. They're constantly being exposed to water, and the ability of water to sort of pull heat. They've actually evolved hydrophobic fur or feathers that basically repels the water. It acts like a repellent. So it keeps the water away from the skin layer out where the fur layer is. As long as that water doesn't touch the skin, it's not going to be able to pull the heat out. So that fur layer is insulated against the water because it's hydrophobic. Again, you have a basic fat molecule like a saturated fatty acid or an unsaturated fatty acid. And you can have fats that are everything from those simple fatty acids to triglycerides. See the upper right, triglyceride, it's three glycerides basically, triglyceride. We have phospholipids, kind of two long chains there, and we'll look a little bit later at how important phospholipids are. Steroids actually fit into the category of being a lipid. Steroids that, again, our bodies need for their survival. Triglycerides you might have heard of if you've ever had a cholesterol screening. The level of triglycerides is one of the indicators as to how well your body's doing with respect to processing these fatty molecules. Of course, when we look at the actual cell and the parts of cells, we'll look at the cell wall. And again, you can see on this on this diagram of a cell wall, you see something at the bottom right labeled lipid bilayer. You see these little things that have heads and two little tails coming off of them. Some are oriented this way and some are oriented this way, the head and the tail and head, and they're oriented opposite each other. That's basically every cell wall in our body has these two layers of lipids, and they're phospholipids. They have phosphorus in them as well. And so they form the cell wall of every cell in our body. And we'll look at that a little closer later. But so fats or lipids play a very important role in our bodies because certain types of lipids make up every cell wall in our body. Again, if you pay attention to your diet, you read labels and such, you undoubtedly hear about saturated fats versus unsaturated fats. Saturated fats are typically the ones that we consider to be less healthy, things like butter, for example. Now, again, there's nothing wrong with butter unless you eat a stick of it a day. Unsaturated fats like olive oil, for example, have your body manages them better. They don't tend to accumulate in your cells like the saturated types of fats do. And again, the difference in these two types of fats is just simply a bending of a molecule. Notice the two diagrams of the molecules are identical, except for the part at the very bottom where it shows the saturated fat basically is oriented straight, whereas the unsaturated fat has a bend in one of the bonds between the molecules that cause it to bend. And that simple difference creates different properties. Unsaturated fats tend to be liquid at room temperature, whereas saturated fats tend to be solid. That's a pretty simple way to know if you're dealing with a saturated or unsaturated fat, whether it's solid or liquid at room temperature. And again, we're not going to get into kind of all the nutritional aspects. It's more or less just to introduce you to some of these ideas. And it's actually during the hydrogenation, the adding of hydrogen, that the orientation is changed, which changes the chemical properties of the saturated and unsaturated fats. It's that hydrogen that's indicated in red when that hydrogen is added in the chemical bonding, which is hydrogenation, adding of hydrogen, that either causes that molecule to stay straight or for it to bend. So something simple like that changes the chemical property. The only thing I'll briefly touch on regarding, say, health and diet is that certain fats are bad for you if you ingest them in too high quantities. And now it doesn't mean that everybody is going to have the same problems. It just totally depends on genetics and how much you exercise and how your body processes these things. But basically, these triglycerides are circulating in your blood. And the saturated fats and the ones that are called trans fats tend to form clumps much easier than the unsaturated fats. And so when they start forming clumps, they can form these things called plaques that can result in atherosclerosis, which is a hardening of your blood arteries. So when these things start clumping together, they start narrowing your arteries, so less and less blood is getting passed. And then in a worst case scenario, like the bottom of those three images, the blood vessel actually gets totally clogged. And then that can result in heart attacks or other similar types of problems. It can be treated and surgery or diet or drugs, but basically it's these triglycerides that are circulating there. And so if your triglyceride level is too high, via a blood test, it might indicate that you could have these types of problems down the road. Now, again, looking at cholesterol, I know people, I have a brother-in-law whose actual cholesterol number is way above the normal range. But when you look at the triglyceride level, and his are really low, you look at the high-density lipoproteins versus low-density lipoprotein ratio. And so those things can offset a high cholesterol number. Our bodies need cholesterol. Every cell has cholesterol in our cell wall. We'll look at that later. But again, it's how your body processes it. So every one of us is different in how we react to things. Some people can eat a much higher fat diet and never have any problems. Other people can be completely the opposite. They don't have a very high diet in fats, and they can still get some of these problems. But again, it's all about moderating everything. Third molecule, macromolecule, are proteins, and these are the most abundant molecules in our bodies. Again, our muscles are made primarily up of protein. It's the protein we typically think about eating when you eat a fish or a chicken or a piece of beef. A lot of what you're eating is actual protein. Fish are relatively low in fat compared to beef, but again, the muscle fiber itself is made primarily up of proteins. So we're eating protein that way. And because a lot of our body weight is muscle, obviously then that means that a lot of our cells are composed of proteins. What also fits into the category of proteins are enzymes, hormones, and amino acids, things that make up our genetic material, for example. Notice it says about 20% of our body is protein. So about 20% of our body weight is protein. Other molecules make up lesser percentages, and then of course a lot of our body's water as well. See some examples of amino acids here. Again, notice they're all made up of a central carbon, so they all have that in common. They're bonded to an amino group. So if you look in the upper left where it's showing the fundamental structure, got the carbon in the middle, the amino group, which is nitrogen and two hydrogens, a carboxyl group, which is a carbon, two oxygens, and a hydrogen, and then a hydrogen atom. And then there's this R group, which can be a bunch of other types of molecules, but those other four things are all common: carbon molecule, amino, hydrogen, and carboxyl group. And then again, the addition of that R group can make all of these different types of amino acids that our bodies need. Our DNA is composed of a whole bunch of stuff that we'll look at later, but basically polypeptides and basically proteins. You look at the structure of the double helix, which again we'll look at a little more detailed in another chapter. But you've got these bases that form kind of the rungs, the spiral staircase. Basically, if it weren't for protein, basically DNA wouldn't exist. So it's important to realize as well that aside from protein being important in our diet, we think to maybe build muscle, protein's also important because it builds our genetic material. It builds our DNA. There are other macromolecules, obviously lots of other stuff on ingredients, although most of them are going to fall into one of these basic categories of macromolecules. All right, so again, what I would suggest is that you, after you've watched these lectures, that you have the PowerPoint slides to go back and look at, to review, especially when you're getting ready for your each of your quizzes. I've got the key terms, which are in the end of each chapter of each book, each chapter of the book, but also I've got that separate file in Canvas. Unfortunately, I guess I don't look at it as unfortunately, but there's going to be some memorization necessary in this class because, again, there are lots of terms for you to study. So use the terminology, use note cards if that helps you. There are lots of sites you can go to where you can find note cards that you can print or even just study online. If you know anybody else that's taking the class, and you get a little study group together, quiz each other on terminology, that would be one of the best ways probably to study for the class. And remember that you have the book, you have your notes to utilize when you take the quizzes. But with that said, if you don't study for the quizzes and you just assume you can look up everything in the book, you're going to run out of time. So study for this class as if you were going to have to take a quiz without your book. Given that you do have your book and your notes and anything else in the world, but you have a certain limited amount of time to take the quiz. So make sure you're preparing. Once we've gotten through the first quiz, you can assess how you're doing. If you have any questions or want any advice maybe on how to study or something you could do different, then by all means send me an email. I'll be happy to try and help you out. So that ends the lecture on Chapter 2.